Got it? A little bit louder? A little bit louder now. What? Okay. Hi, John.
Hey, Bob.
How are you?
I'm doing fine.
Thanks for coming down yesterday. Really appreciated it.
Had a great trip. I enjoyed visiting the discussion section, seeing Daisy at work.
Yeah, they got a little intimidated when you came down. I think Daisy completely lost her memory when you entered the room. But then it was resurrected when you went back. In any case, what's so funny, Rose? I know. You okay, guys? Steve? Mike? Good. Marvin, you happy? Still nervous like you were 10 weeks ago? Huh? Haven't had an email from you all quarter. I'm shocked. Good. I'm not going to, I have announcements, but I'm not going to put them up because Professor November has done something which, if I touch, I don't know how I'm going to ever get back into. So we'll just leave it as it is. The announcements go very simply and as following. Next week's discussion is on gene therapy. And you have the articles for gene therapy that you got on Tuesday. And so please read those articles. One deals with what cloning means for gene therapy. One article is on obstacles of gene therapy. And another article is on just gene therapy. The article on gene therapy has the figures in it that I used. Thank you, John. Has the figures in it that I used for the lecture on gene therapy. And that article will be very important for your oral exam coming up next week, okay? And then there's some reading in Chapter 11. There's a section on gene therapy in the textbook. Please read it because it's pretty good and it'll complement what you have in the Scientific American articles. So please do that. The next thing is your oral exam coming up, which is a week from today. And that oral exam, as we discussed, will be very similar to the format of the midterm oral exam. Start now. If you started, Albert, organize it. As I've said, you know, organize before you do anything. Where can I access the information? Organize it, and then bullets, okay? You weren't too overwhelmed, were you, Max? When you looked at it? Almost had a heart attack? You did? It's a short exam. I could have made it to 20 pages instead of 2 or 3, you know. What are you laughing at, Dora? You probably learned more about science in this class this quarter than you've learned in the last 15 years of your life. Do you agree?
I agree.
Yeah, right. Exactly. In any case, get on it, please, because it's not something which, as you know, can be done in a second. There was one science development announcement that I was going to give to you, but I'll just relate it to you. I'll show you the picture on Tuesday, and that is that the holy grail of rose breeders, and I don't mean rose, but rose breeders, you know, people who breed roses, has always been to get a blue rose. None of you have ever seen a blue rose. Well, they've now engineered genetically a blue rose by taking a gene from an iris and a gene from a pansy and then adding another construct, this thing called RNAi that I talked to you about, 3 independently put in different genes, and they've been able to engineer dark blue rose, light blue rose, medium blue rose, and all shades in between. And so look in your favorite florist in the coming years for blue roses. Any questions about the oral exam coming up next week? Elaine, you happy? Any questions, Harumi? Looked kind of confused in discussion yesterday. Were you sleeping? Were you in a haze? Or what was going on? Daisy called on you and went, huh? Remember that, Daisy? Huh? You were? Yeah, maybe more awake than I was a few hours ago before class. Faria, how are you?
Doing well.
Are you ecstatic that you only have one more week of Bob Goldberg?
No, I'm actually really sad.
Probably going to be a party at the end of next week, I'm sure. No more quizzes, no more being called on in class. Everyone can slip back into their original anonymity and be in classes, sit in the back row, look at their cell phones, bring in coffee, munch on pastries, and go back to the old ways of doing things. Is that right, Eden? I don't... Listen, you got one more quarter with me. You haven't seen anything yet. If you think this was a lot of work, multiply it times 1,000. That's what you're facing next quarter. Okay, we have a wonderful guest speaker here, Professor John November. And as I've said with some of the speakers, John just arrived on campus in 2008, and as I said many times, I arrived in 1976, so what does that say? He's probably young enough that he could probably be my son or something like that, even though I don't have any sons or daughters, but as all of you know. But the fact of the matter is, John is really, and I'm not going to embarrass you, John, but I'm going to try. John is really a rising star. Remember, I told all of you guys that on Tuesday. And he's really one of the world's experts, absolutely one of the world's experts in the field of using the human genome to trace human ancestry. And in many respects, he's been doing and has been doing a lot of the pioneering work that we talked about, and really getting down to the kinds of resolution that these ancestry companies that you're writing about for your quiz this week couldn't even dream of. And so John's going to talk to us about that, and feel free, as I said on Tuesday, to ask a lot of hands-on questions about how these things are done and how samples are gotten and where they're stored and how they handle all these different samples and just the nitty-gritty stuff. Just to tell you a little bit about John, correct me if I'm wrong, he got his PhD at the University of California, Berkeley, and was interested in population genetics and decided then to do a postdoc at the University of Chicago, which has some pretty luminary people who work in the area of human genetics. And after doing a very wonderful series of work of tracing human ancestry, mainly in Europe, correct? And developing techniques to do it. And I should say that he's really quite a mathematician and statistician, which is what you have to do looking across all these millions and millions and hundreds of millions of nucleotides and SNPs across the human genome. UCLA decided that we were going to entice him here with hopefully a wonderful package that made him very happy, because most of the young professors that we get at UCLA are sought after by many places around the United States. And so I'm going to turn the floor over to Professor John November, who's in the Department of Ecology and Evolutionary Biology, and whose office is in the Life Sciences building. Well, 5th floor, right? 4th floor, right above us, a couple floors up. And he's going to talk about tracing human ancestry using SNPs and all other things in the human genome. And got it? A little bit louder? A little bit louder now. What? Okay. Hi, John.
Hey, Bob.
How are you?
I'm doing fine.
Thanks for coming down yesterday. Really appreciated it.
Had a great trip. I enjoyed visiting seeing Daisy at work.
Yeah, they got a little intimidated when you came down. I think Daisy completely lost her memory when you entered the room, but then it was resurrected when you went back. In any case, what's so funny, Rose? I know. You okay, guys? Steve? Mike? Good. Marvin, you happy? Still nervous like you were 10 weeks ago? Huh? Haven't had an email from you all quarter. I'm shocked. Good. I'm not going to, I have announcements, but I'm not going to put them up because Professor November has done something which, if I touch, I don't know how I'm going to ever get back into. So we'll just leave it as it is. The announcements go very simply and as following. Next week's discussion is on gene therapy. And you have the articles for gene therapy that you got on Tuesday. And so please read those articles. One deals with what cloning means for gene therapy. One article is on obstacles of gene therapy. And another article is on just gene therapy. The article on gene therapy has the figures in it that I use. Thank you, Jack. Has the figures in it that I used for the lecture on gene therapy. And that article will be very important for your oral exam coming up next week, okay? And then there's some reading in Chapter 11. There's a section on gene therapy in the textbook. Please read it because it's pretty good and it'll complement what you have in the Scientific American articles. So please do that. The next thing is your oral exam coming up, which is a week from today. And that oral exam, as we discussed, will be very similar to the format of the midterm oral exam. Start now. If you started, Albert, organize it. As I've said, you know, organize before you do anything. Where can I access the information? Organize it, and then bullets, okay? You weren't too overwhelmed, were you, Max? When you looked at it? Almost had a heart attack? You did? It's a short exam. I could have made it to 20 pages instead of 2 or 3, you know. What are you laughing at, Dora? You probably learned more about science in this class this quarter than you've learned the last 15 years of your life. Do you agree?
I agree.
Yeah, right. Exactly. In any case, get on it, please, because it's not something which done in a second. There was one science development announcement that I was going to give to you, but I'll just relate it to you. I'll show you the picture on Tuesday, and that is that the holy grail of rose breeders, and I don't mean rose, but rose breeders, you know, people who breed roses, has always been to get a blue rose. None of you have ever seen a blue rose. Well, they've now engineered genetically a blue rose by taking a gene from an iris and a gene from a pansy and then adding another construct, this thing called RNAi that I talked to you about, 3 independently put in different genes, and they've been able to engineer dark blue rose, light blue rose, medium blue rose, and all shades in between. And so look in your favorite florist in the coming years for blue roses. Any questions about the oral exam coming up next week? Elaine, you happy? Any questions, Harumi? Looked kind of confused in discussion yesterday. Were you sleeping? Were you in a haze? Or what was going on? Daisy called on you, went, huh? Remember that, Daisy? Huh? You were? Yeah, maybe more awake than I was a few hours ago before class. Faria, how are you?
Doing well.
Are you ecstatic that you only have one more week of Bob Goldberg?
No, I'm actually really sad.
Probably going to be a party at the end of next week, I'm sure. No more quizzes, no more being called on in class. Everyone can slip back into their original anonymity and be in classes, sit in the back row, look at their cell phones, bring in coffee, munch on pastries, and go back to the old ways of doing things. Is that right, Eden? I don't. Listen, you got one more quarter with me. You haven't seen anything yet. If you think this was a lot of work, multiply it times 1,000. That's what you're facing next quarter. Okay, we have a wonderful guest speaker here, Professor John November. And as I've said with some of the speakers, John just arrived on campus in 2008, and as I said many times, I arrived in 1976, so what does that say? He's probably young enough that he could probably be my son or something like that, even though I don't have any sons or daughters, but as all of you know. But the fact of the matter is, John is really, and I'm not going to embarrass you, John, but I'm going to try. John is really a rising star. Remember, I told all of you guys that on Tuesday. And he's really one of the world's experts, absolutely one of the world's experts in the field of using the human genome to trace human ancestry. And in many respects, he's been doing and has been doing a lot of the pioneering work that we talked about, and really getting down to the kinds of resolution that these ancestry companies that you're writing about for your quiz this week couldn't even dream of. And so John's going to talk to us about that, and feel free, as I said on Tuesday, to ask a lot of hands-on questions about how these things are done and how samples are gotten and where they're stored and how they handle all these different samples and just the nitty-gritty stuff. Just to tell you a little bit about John, correct me if I'm wrong, he got his PhD at the University of California, Berkeley, and was interested in population genetics and decided then to do a postdoc at the University of Chicago, which has some pretty luminary people who work in the area of human genetics. And after doing a very, very, very wonderful series of work of tracing human ancestry, mainly in Europe, correct? And developing techniques to do it. And I should say that he's really quite a mathematician and statistician, which is what you have to do looking across all these millions and millions and hundreds of millions of nucleotides and SNPs across the human genome. UCLA decided that we were going to entice him here with hopefully a wonderful package that made him very, very happy, because most of the young professors that we get at UCLA are sought after by many, many places around the United States. And so I'm going to turn the floor over to Professor John November, who's in the Department of Ecology and Evolutionary Biology, and whose office is in the Life Sciences building. Well, 5th floor, right? 4th floor, right above us, a couple floors up, and he's going to talk about tracing human ancestry using SNPs and all other things in the human genome. And so, John, thank you so much for agreeing to do this. I know that you're new at this kind of thing, having not even taught a class at UCLA yet. You're going to be a good introduction. So thank you very much. The floor is yours.
Okay, great. All right, thanks so much, Bob, for that introduction, and it's a tough act to follow. It seems like you have a really great rapport with the class. Let's see, I'm going to be talking about human ancestry and giving a kind of broad overview of the field of what we know in human population genetics. That's my field, what I've been studying, and broadly I study population genetics in general. The field of population genetics is really the study of genetic variation. And why does it exist in nature? Why isn't every organism in every species exactly identical? Why isn't there just one most fit type that is sort of identical in any species? There's always diversity. And there are many reasons why that diversity arises. There are many ways in which it changes through time. And that diversity has outcomes in terms of the phenotypic diversity that we see in all organisms, and their ecological performance, and in how they evolve over time. So it's a very a field that has very broad questions, and for a long time though it's been very data limited, in that They're very sophisticated models of how genetic diversity changes through time, but we didn't have access to any of the sequence information to actually study how genes change through time. And now with kind of these revolutionary technologies, we're finally being able to look at some of that genetic diversity up close. And then one of the best places to do it is in humans. So of all species, humans are the best studied in terms of our genetic diversity. And that's mainly driven by the biomedical interest in that genetic diversity in humans explains a lot of the differences in disease predisposition that we see. So in some ways, humans are a model, sort of like a model organism within population genetics. They're a particularly good species to study. But beyond that, they're also fascinating because of we are humans, and it's always interesting to understand our own history and how and our ancestry. So let's see. I need to get this started. Okay, great. So some of the themes I'm going to cover today are global patterns of human genetic diversity, tracing our most ancient ancestry. I'm going to talk a little bit about something that's known as the clines versus clusters debate regarding human population genetic diversity. We're going to look at some within-continent patterns of diversity, talk a bit about personalized genomic ancestry inference. These are the kinds of services that are being offered by many companies. We'll kind of ponder a little bit what really is ancestry when we talk about ancestry inference. Talk a little bit about the idea of admixture and what admixture is and the idea of chromosome painting. And then finally, I want to talk a little bit about natural selection in humans and the patterns of human genetic diversity, how it impacts patterns of human genetic diversity. So just to start us off, if you look at the fossil record in humans, and these are fossils from East Asia, you see, let's see, did I have a pointer? Right there. Okay, great. So you see the appearance around 1 million years ago of these fossil lineages that are represented with orange lines here. And these are fossils of Homo erectus. Homo erectus, you might guess from the name erectus, was one of the first humanoid ancestors of modern humans that actually stood straight up. And not only did they stand straight up, they left Africa, where they originated, and spread out across the globe. And a major question in human evolutionary biology for a long time was, we know that this Homo erectus species has spread out across the globe. Did Homo sapiens, the Homo sapiens sapiens, the very modern humans that we are today, are we all descendants from Homo erectus directly, so that modern East Asian individuals living today are a direct ancestor of Homo erectus that emerged from Africa 1 million years ago? Or is it that Homo sapiens sapiens arose separately in Africa and then spread out across the globe in a second expansion? Okay? And part of that puzzle arises because you have these lineages of fossils that exist and change through time, and then there's a big gap from 100,000 years ago to 40,000 years ago where there's no fossils. And then around 40,000 years ago, you start seeing all of these Homo sapiens sapiens fossils. And it's not clear whether this is just because fossil records have gaps. It's often not, there always aren't perfect conditions for preserving fossils. And so you can't be sure if this is just because the conditions weren't good enough to leave fossils, or if in fact Homo erectus and the Homo sapiens that followed them died out and then were later replaced by very modern humans. Okay, so we have this open question. And this is kind of drawn out in these competing models of human origins, of the early human origins. So we have these different lines representing different continental regions of the world, with Africa, Europe, Asia, and Oceania. And this first model, Model A, is the model of a recent out-of-Africa origin, where we have a lineage from Africa that spread out across the world that represents Homo erectus, that then died out everywhere and then was replaced by Homo sapiens sapiens. The second model is that Homo erectus left, but there was some slight amounts of contribution to modern humans. A third model is that you had Homo erectus never died out, and there was just persistence of the lineages and an evolution towards Homo sapiens sapiens communally across the world via shared gene flow. So this is called the shared multi-regional model of evolution. And then the last model, D, is of independent multiple origins, that modern humans arose separately in each of these different continental regions. So you guys have these clickers, and I guess this is a good time for me to test how this thing works. I think probably you guys got an introduction, might have a good solid guess at what the answer should be from Bob's lecture, but just to see how this thing goes. So how's it work? They click away, oh, and then I get to see the answer here or something? Oh, it's like on a different...
Yeah, it's on a different little jigamaboo, right.
Here,
So
turn around, Bob.
Unless we mirror.
Where is it?
There it is, right up there. Start.
Start. Okay.
I'm so sophisticated here that I don't even know what is done with my computer.
Okay, so then you...
And then you let them go for there's about 25 to 30 responses. You let it go, and then you do stop, and then you do display.
Okay. Great. So you guys are clicking away. Now, the people in Davis get to click away?
Well, we'll ask them when they're done.
Okay. Okay, is that good? 24?
Stop.
Stop. Display.
There you go.
Okay, you guys are pretty sharp. What do we got? We got E. I didn't know that could be an answer. All right.
Means they don't know.
And then recent
That's
with
Albert
minor
pulling your
contribution.
leg. This guy Albert is always the one that's pulling everyone's chain.
Okay, great. So let's go back here. Yeah, you guys
So
know
what's the
going
reason
on.
for your answer, Rose? Yeah.
Well, I said recent out of Africa with minor contributions, but wasn't there some sort of like, in some of the articles we read, there were different like conflicting things about whether or not it's entirely out of Africa or out of Africa with some like Neanderthal or like whatever contributions where it's just kind of like there's a tiny little snippet of our DNA that's like that.
That's right, yeah. So you've hit on a controversy. And right now, it's completely an open question. There's some hints of a minor contribution, but it's not, we can't be sure. And let's see, I wonder.
How did you vote, Dora?
Okay.
I said A
Why?
because we talked about how the people around the world originated in Africa because Africa had like the most diverse genetics.
They were most in terms of genetic diversity, right?
Yeah.
What about you, Faria?
Same.
Same?
Yeah.
Ryan must have done a really good job with his discussion yesterday, huh?
Yeah, he did. Of course.
That's
Amazing
great.
as usual.
Who said multi-regional origin? Who voted for B? Someone did. C. Who was the one that put E down? Okay, go ahead, John.
Okay, great. So, right, and you know this because you've been shown the mitochondrial DNA tree, which shows this is the history of, it's a guess, right? We infer this tree that connects all the individuals that we've sampled today, and we make a guess at what this tree looks like, and that tree always shows individuals from outside of Africa having ancestry that's around 50,000 years ago. And then you have to go further back in time to have ancestry with African lineages, and then those go back to around 170,000 years ago. And that time period is really crucial. It's around 200,000 years ago, whereas if it was the multi-regional hypothesis, we would trace all of our ancestry back to around 1 million years ago, because that's when Homo erectus left Africa and colonized the world. Okay, so that time date's really important. And part of what causes the controversy about whether there might be some minor amounts of ancient ancestry, is that if you look at most genes in the human genome, they will have dates of common ancestry that are in the range of 200,000 years ago, 300, 400,000 years ago. But now and again, you get a few genes that have ancestry that goes back 2 million years. And so those genes could be genes that some ancestors, say I have one of these genes takes a very long time to connect, have common ancestry with the rest of, say, everybody else in this room. It might be because one of my ancestors at that particular gene locus was from a Neanderthal, and then you have to trace back to when Neanderthal and humans were common ancestors to actually have it connect, which would be more like millions of years ago instead of hundreds of thousands of years ago. But there are debates about whether you could explain those patterns by having, within Africa, different subpopulations. So that would also push these ancestry times back. Okay, so, so you have this mitochondrial Eve that existed in the recent past, and this tree shape is consistent with the out-of-Africa origin. And what I want to talk about is that's the mitochondrial DNA evidence. I'm going to kind of emphasize a lot of my results based on single nucleotide polymorphism results, because that's really, there's been a whole wave of studies within the last year that have been presenting results from these new microarray-based SNP, high-throughput SNP genotyping technologies. And a number of these have been based on a set of samples from around the globe known as the Human Genome Diversity Panel, and mainly a collection that was spearheaded by researchers at Stanford University in collaboration with a French group of researchers. And it includes 52 populations from around the world, and these are their geographic locations. And it's not a perfect sampling of the world. There are big gaps. There's no samples from Australia, New Zealand, none from, there are lots of interesting people living in the Pacific Ocean. There's no samples from India. It's sort of sparse within Africa, sparse across Central Asia, sparse across the Americas. But it's the best studied samples that we have. And it's kind of a reference data set that as each new technology comes out that allows us to study genetic diversity in a new way, it's being applied to the same sample. And so we keep getting a finer and finer scale picture of human history just by revisiting the same group of samples. And so, as I just mentioned, the main thing I'm going to be talking about are single nucleotide polymorphisms. And this is a slide taken from one of Bob's lectures earlier. Just to remind you, these are locations in the genome where you have two different base pairs, and so that you have a single nucleotide position that is polymorphic. It's variable. And these SNPs taken together define haplotypes, which are the combinations of SNPs along a particular chromosome. And you can even make things really easy for genotyping by picking out a few diagnostic SNPs that would, just with these three, we can tell what the whole pattern was across that larger set of SNPs and define just these short haplotype tags.
You guys remember all of this? We went over this, remember? Okay. That you have these diagnostic SNPs, as John said, that represent the different haplotypes, and I know you went over haplotypes yesterday again in discussion, so you should be familiar with this. And remember the DNA chips, we went over that in excruciating detail, so I think you're all comfortable with these things. Remarkably so, though.
Great, and feel free to interrupt at any point. So there are these SNP chips, and so what you can do then is look at genetic diversity now. Instead of just looking at the mitochondrial DNA, which is one small molecule, it's just 16 kilobases of DNA, and even most of that doesn't vary. Most of the studies of mitochondrial DNA are based on a very short part of it that's hypervariable. So now we have 650,000 SNPs, and we can reconstruct a tree based on allele frequencies for all of these populations around the globe. And what we see is this really clean pattern where we have Oceania, the populations from Papua New Guinea and Melanesia, the American, Native American populations, East Asian populations, populations from Central Asia, Europe, North Africa and the Mideast, and Sub-Saharan Africa. And you still have this tree structure that supports an out-of-Africa hypothesis for the origins of modern humans. A really striking result, too, is if you look at a statistic called haplotype heterozygosity. So this is a statistic that calculates how often, for a particular haplotype, each individual is heterozygous. Okay? I have two haplotypes. Each of us have two haplotypes at any particular gene region because we have one haplotype that's inherited from our father and one from our mothers. And we look at how often are those the same or different. Okay? And if we're from a smaller population with less genetic diversity, our mothers and our fathers would have passed us the same exact haplotype copy. And so our haplotype heterozygosity would be 0. If it was different, our heterozygosity would be 1. And we're going to take an average of those 0 and 1 values across all the individuals in our sample, and we get a number. And that number, just more intuitively speaking, represents genetic diversity. Higher haplotype heterozygosity means more diverse. And what you see is this nice decay of diversity with the distance that you travel from Addis Ababa in the Rift Valley of Ethiopia. Okay? This is a putative origin of early modern humans. This distance is calculated using waypoints. Okay? Because if I calculated the distance between say, Papua New Guinea and East Africa directly, I'd be crossing the Indian Ocean. So the distance we use here actually goes through going only along land routes. Okay? And what you see is this nice decay with distance, which fits a model of humans leaving in small groups to colonize a new area. And when they leave in a small group, they're only going to take a subset of the diversity that they had before, right? And so you'd think that the groups that are just outside of Africa are going to have lost some diversity. But then when you then go to colonize the next location, you're taking a subset of diversity, so you have even less diversity. And you keep moving until you get to the Americas, where Native American populations have some of the lowest levels of genetic diversity in humans because they have sort of been through so many of these founder events, including crossing the Bering Strait around 18,000 years ago, it's guessed. Okay? So all of this fits a model of the out-of-Africa expansion in much more detail than just looking at that mitochondrial DNA tree. And this is just a sort of picture to show what these waypoint distances look like of routes going out across the world and crossing over the Bering Strait to get to the Americas. And when you use these kinds of distances, you see this really nice pattern. So it's really cool that you can actually kind of see how humans expanded out of one location and how the diversity tailors off as you get further and further away from that central location. And you can even ask, if I had to place one area to measure that distance from to get the best fit line of decay and say pick the best location in the world for getting a nice decay like that, the best location is in East Africa. Okay, so if I move that dot up into, say, near Moscow, I wouldn't get that nice decay pattern. It's really about having that origin in East Africa. So this is just a figure to emphasize that kind of decay of diversity. Here's Africa, where it's very diverse, and then you're going to have lower diversity just outside, and then even less as you go further and further away. Okay, so that's somewhat of a review for you guys, but just looking at modern SNP data for doing it. But at a global scale, these genome-wide diversity patterns are broadly consistent with the single recent origin of modern humans in Africa. I also didn't talk about the Y chromosome data, but that data also supports a recent origin. And I did mention that autosomal genes. So if you look not only at mitochondrial DNA for when was the time of the most recent common ancestor, but you also look at genes in the, on your autosomal chromosomes, right? In the rest of the genome. You'll also see these times of most recent common ancestry that are very young, in the range of 200,000 years ago, and which are much too young for there to have been an ancient origin of humans. But as I mentioned, there are these very few small number of loci that show much more ancient TMRCA dates, the time to the most recent common ancestor. Okay? And that leaves open this debate about maybe there's evidence for rare ancient contributions from archaic humans. And I didn't get into it, but that's where this Neanderthal genomics is so exciting, is the idea that potentially by looking at the Neanderthal genome from sequencing DNA from these fossil specimens, we might be able to find evidence of where Neanderthals mated with humans in Europe when they actually overlapped there. But that's all still very, very new research, also very controversial, because you can imagine if you're studying a Neanderthal DNA and you claim that you have evidence for sharing with humans, it could almost always be explained by some contamination in the test tube from the humans that are in the lab getting into the sample. And so there's been a lot of back and forth in that literature of people saying, look, we've got evidence for mixing between Neanderthals and humans, and then other groups saying, look, with just the littlest bit of contamination, you would get that result anyway. So it's kind of a hot research area right now to straighten those issues out. Okay, so now I'm going to shift to talking about this Kleins versus clusters debate. And this is really driven by this, started by this really influential figure. This was published in 2002, and this was on that same set of populations, the same set of 52 populations, the Human Genome Diversity Panel. And it was before even looking with SNP loci. There was a large survey of around 700 microsatellite loci. Did you guys talk about microsatellites? You talk about VNTRs? Yeah. Yeah. So most of these are STRs. These are short segments of a genome that have a set of letters, maybe 4 letters, that are then repeated a number of times, and individuals in a population will vary in the number of repeats they have. And so if you collect 700 of these STR loci genotypes for all of these 52 populations of individuals, and you use a clustering method, a population genetic specific clustering method known as structure, and you ask it to assign each individual a set of proportions of how much of its genome is from any of K populations. So here we're running it with K equals 2. And each of these bars represents a single individual. And the method is going to tell us that, the result it's giving you here is that it's saying all of these individuals are 100% of the orange population, and all these individuals are nearly 100% of the purple population, and I'm going to say these individuals are sort of somewhere in between. Okay? But it has no idea. It's an unsupervised method. You're not telling it anything about what populations you're putting into the analysis, and you're just asking it, create some groups for me. And what really struck people is, so this is doing it with saying, give me 3 groups, give me 4 groups, 5 groups, 6 groups. And you can see how when you say do it with 4 groups, without knowing anything about the geography of the world, this method groups the individuals who are from Africa, groups the individuals who are from Europe, the Mideast, and Central South Asia, okay? And then groups East Asian samples, the Oceania samples. Oh, I was on K equals 4, wasn't I? Okay, so it groups East Asia and Oceania, and then it groups the Americas. Okay. And you can keep going, and you at sort of K equals 5, you break out even Oceania from East Asia. And then at K equals 6, it breaks out this one very specific group known as the Kalash that have been very isolated. So they kind of emerge as a distinct group. And even though the scientists who wrote this paper didn't make this claim at all, some people saw this and said, well, this is genetic evidence for race. We've got these very clean-looking clusters of groups that are genetic and that have completely been defined by the data. And that got a lot of people a bit thinking again about the genetics of race, because for a long time population geneticists have been saying there isn't much reality to race because all the early data is showing that most of the variation in humans is shared between different groups around the world. And this picture sort of creates that a different impression. And this is revisiting that same analysis, but now with 650,000 SNPs. And you can see with more data, you can even get finer scale resolution. So that you see before, you're seeing kind of this big group of Europe, the Mideast, Central South Asia. With more data, you get now a sort of European cluster, a Central South Asian cluster. But you still have the main geographic structure. And then the Mideast looks like an area where you have some component that is unique to the Mideast, but then also some influence of European ancestry and Central South Asian ancestry. Okay. And this is oftentimes, a lot of this debate is about how you represent genetic diversity, because these images really make it look like there are these distinct clusters. Whereas if you look at this image from the very same data, things don't look so dramatically differentiated. So what this image is a plot of genetic distance between different populations as a function of geographic distance. And here what you see is that differences between populations appear to increase really smoothly with geographic distance. So you have two different pictures. One that's very clustered of distinct groups, and then this one is sort of the clinal picture. Clines are smooth gradations. And this gives you the impression that all of human genetic diversity is driven by geographic distance.
John, in the Rosenberg paper, we actually talked about that paper, but you don't know about it. We discussed that paper from the point of view, I think it was a Rosenberg paper, where they said really that about 95% of the genetic variability was shared between all groups.
That's right.
And this was at 5% that they could classify into these geographical distinctions. Am I correct about that?
Right, that's exactly
So the plot that you would see up here and what John is showing you is the differences between the different groups. That's what you're getting. Not the similarities, but the differences.
That's right, yeah.
So keep that in mind.
Yeah, and I'm going to come back to that point, but that is absolutely what's going on, and part of what causes the confusion is that some of these representations of data don't tell the whole story. But regarding this particular Clines versus clusters, one of the thoughts about this is that the reason you see the clusters is because of gaps in the sampling. So there are no samples in this area of the Tibetan Plateau, so maybe that's why you see a cluster of East Asians versus Central South Asians. There's kind of a gap in the sampling here. So everywhere where you could put a cluster boundary is also where there's a decent gap. But it's also where there are some pretty major geographic barriers, like the Tibetan Plateau. You can imagine being a barrier to human dispersal. The Bering Strait, you can imagine being some kind of barrier to human dispersal in the Sahara. And the Mediterranean. So you have this kind of, it's difficult right now with this sample to tease apart what is due to non-uniform sampling Okay, because even if things are very smooth across a gradient, and I only sample two ends of it and not the middle, of course it'll look like I have two groups. So that's where that's one potential explanation for it. And that's what people who hold a clinal view, how they would explain it. Individuals who have the cluster view of human genetic variation would say these geographic barriers have led to slight differences that then this method can pick up on. Okay. But either way, and this is the point Bob was making, it's important to remember that variation is mostly found within rather than between groups. And this shows up in the same data set as well. It's kind of unfortunate that they cut the axis here because you would really be able to see it much stronger, that in each of these cases here, it's 10% or less. That's right, you were saying 5% from talking about the Rosenberg paper. That's this particular data set. At SNP variation, there's slightly less shared, but it's still the bulk of it, 90% of the data, is shared between groups, and it's only this last 10% that really differs between groups. Okay.
So let me ask you a question, John. Is everyone understanding what John is saying here in terms of the implications of this? Because it's significant. If there were discrete clusters, what would that mean conceptually as compared to a cline with respect to the diversity, or let's say the migration of human populations and or relationships between human populations?
What would it mean in terms of the implications for history?
Yeah.
Yeah, so...
Does everyone understand that question? I mean, what would that mean if there were discrete clusters with respect to where we came from, and how would that maybe be translated into this race argument?
Right, so the clusters, if they were real, would reflect that there's been larger amounts of time separating that human groups have been in isolation from one another. And this plays into the race argument. This is getting a little bit out of my field, but in that it gives room for the biology for humans to be quite different biologically. Okay. Whereas if it's clinal, there's more of a smooth gradient, there's no clear boundaries, and there's no way you can make any of these distinctions. Now in reality, it's actually, I think that's how it plays into people who think about race in these simple terms. The fact is, even if the clusters model were true, the clusters could be so that these geographic barriers actually have led to differentiation that's a bit enhanced. If the clusters are very young, then most of the variation is going to be shared between all human groups anyway. So it's just like a slight bump in differentiation of overly similar groups. Yeah.
So I have a question about the Klein data. Is that like a different data set than the clusters data?
It's actually the same data.
So I guess I just don't understand how the same data set can show two different theories.
So, yeah, I wish there's one figure from another paper that shows it a little bit better. So
I just kind of feel like if you get cluster data, like wouldn't also the genetic distances be sort of clustered as well?
No, so I think that what's going on is that the geographic distances compare, they compare groups that are, actually this specific plot too, okay. The comparisons are kind of comparisons that are going across some of these geographic barriers and between them. And so you get, for any geographic distance, you can get some vertical spread here that kind of obscures that. So some of the comparisons between, so each one of these is a pairwise comparison, right? And all of the ones that are going to be across geographic barriers are going to sort of be up here. Because for a given geographic distance, you've got a little more genetic divergence. Okay. And all of the other ones are going to be sort of down here. That actually sort of you're right. So if I had if I could draw it like,
Go over the whiteboard over there.
Where is it?
Right over there.
Over there. Okay. Okay, so say we had two populations that have there's some kind of geographic barrier that's led to some clustering between them. Okay, and say the clustering. Okay, and then we're going to do comparisons of how different. Their populations within these larger groups. Okay. Okay, and I'm going to do pairwise comparisons. Okay, so say I take that comparison and I look at how much genetic distance there is, and say that's that distance. So I'm going to get a value that's sort of low. Okay, I take the same geographic distance, but I go across the boundary. I'll do that. I'll get a point here. All of the comparisons between clusters will pop up here, high. All the ones within clusters for a given geographic distance will be low. Okay, and if we do this and say I have between cluster ones that are farther apart, okay, so we go farther apart, and there'd be points up here. And basically, you'd expect to kind of get two lines. Yeah, this is a good point. It's a great point to make. You'd actually expect to get two lines where geography matters in both cases, but the barrier is leading to a big difference between within versus between cluster comparisons. But what's going on in the human data, to the extent there are clusters, or the barriers are there, they've got to be very weak because this distance is not very big. And there's actually another plot in another paper that that talks about the Kleins versus cluster debate and tries to make this point, that if you look at it and stratify it, you see, like of that same plot, if you kind of divided it up of ones that are across the barriers versus within, they're just slightly like that versus that. Okay, and so that paper argues that it's a little bit of both because you do see a shift upwards, but it's just not very strong. So that Kleins are really playing a major role, but the barriers do seem to matter too. And if it was just a sampling artifact, it wouldn't matter. You wouldn't get that shift upwards. Yeah.
Does everyone understand? It's a subtle point, but these geographical barriers that we've talked about in class, the oceans and the deserts and the mountains and stuff, really cause these populations to be, reproductively isolated from each other. But it doesn't seem that there's been enough time, as I infer, that you could get large differences genetically between these populations. And I think John is reinforcing what we've talked about in the class, that it's really a gradient of peoples out of Africa in terms of the migration, with probably overlap on the edges if there wasn't any geographical barriers in between to get this differentiation. Julie, go ahead.
So is the potential for the clusters to become further defined or to have the lines separate even more kind of gone now because of modern transportation?
Yeah, I think that's kind of the general thought, is that
exactly.
With modern migration, that any levels of differentiation is kind of quickly going to be erased. Yeah.
We know that humans are 99.9% similar, but I was wondering what percentage of that 1% encodes for genes?
I would guess that it's actually pretty uniformly distributed across genes, so that whatever portion genes are of the whole genome, which is, I don't know the number off the top of my head, but it's small. Most of the gene.
Remember that most of this.
And actually,
Yeah, go ahead.
So two things. It's actually going to be, most of it is actually even going to be in the non-genic part because of selection to conserve the gene regions. Yeah.
So remember, the coding sequences in the human genome are about 0.5%. It's tiny, the exons. But these data primarily deal with non-coding regions throughout the human genome and are not subject to the same selection pressure. And if it was a data set, and John's the expert, I think it was a data set that was, undergoing very, very strong selection pressure, you'd get very different kinds of answers.
Right, right. And so the last part of this, we'll talk about how natural selection plays into all of this. Okay. But these are great points. Is that helping clarify things? Good. Okay. So, okay, and just to kind of add into I think we've gone pretty far in the clines and clusters, and that's really a great point to bring up in terms of clarifying it. But another thing is just kind of the level to which, as you get more data, this is another kind of revisiting of the HCDP data. There were actually two groups that separately used high throughput genotyping to look at the HCDP in the same year. And it was actually interesting. One of them was a student of the other one, but had graduated from the lab and went and did, his own attack on this problem. So you had sort of advisor versus student in a publication race. One paper ended up in Nature and the other in Science. And what this is Noah Rosenberg's group now that showed that as you can even see differentiation within each of the clusters. So if you look more closely, you see more clusters as you get more data. So that sort of says, well, these 5 clusters is kind of an artifact of what scale you look at the data. If I just analyzed the Middle Eastern, North Africa, Middle Eastern proportion, I would see even further subclustering. Okay? So it's not that there are these 5 or 6 monolithic human groups that are completely uniform within them. If you look even closer, you'll see those are divided. And then this is where some of my research plays in, is that I was able to look at a very large sample of European individuals, around 1,400 European individuals from 37 unique populations within Europe. So this is nice because previous samples have always been at this global scale, and I had access to this data that was at a very fine spatial scale within Europe, 37 different groups. And the way that I looked at the data was to use...
Can I interrupt you just a second, John, before you get into that? So what we had discussed in the class, how do you get the DNA from 1,400 different individuals? Who collects the DNA? How does it get cataloged? How do you handle it? I mean, how does just, the nitty-gritty for those of us who haven't thought about ever doing this, and I mean, you go and take cheek swabs of all these, I mean, how's this done?
Yeah, okay, well, I'll tell you, I'm also in the bioinformatics program here, so I'm kind of a computer jockey more than anything else. So for me, the nitty-gritty of the data arriving involves an FTP transfer. So it's not too complicated. But what happened here is this kind of scale of sampling involves a lot of logistics and a lot of different people. And it also involves a lot of money. And so the way that this particular sample came together was that you had different academic groups around Europe genotyping local populations in order to do disease studies. Okay? And 2 of the main groups were in Lausanne, Switzerland, and in London. And these groups kept track of every individual that they genotyped. They asked them, where does your, or for most of them, where do your grandparents come from? Where do you come from? Where do your parents come from? Where do your grandparents come from? And so even though they were sampling in London, they got examples of European individuals from around all of Europe. Example in Lausanne, example, again, individuals from different parts of Europe. And there were other smaller groups involved as well. So that these academic groups had been sampling individuals for individual disease studies. And GlaxoSmithKline is a drug company interested in pharmacogenomics, which you might have discussed. It's the idea of having drug prescriptions that are individualized to certain individuals based on genetic, the particular genetic DNA sequence variants of a given individual. And So GlaxoSmithKline offered to do the genotyping for free for all of these academic groups so that to help kind of facilitate research on background levels of genetic variation in Europeans. And so the benefit to the academic groups is free genotyping, and they can do their disease studies using the most advanced and recent technologies. The advantage of GlaxoSmithKline was to have a view of what genetic diversity looks like in Europe, and that helps them in two ways. One is that the first way is that they work on such a large scale that they're running, say, drug trials in 10 different countries, and they will get adverse drug response events from particular individuals in each of these different regions. So the drug doesn't work, somebody had a really bad reaction, and they want to study the genetic basis of that. Well, if they try to do that, you need to, precisely because there is this subtle levels of differentiation amongst humans, you need to match your cases with your controls. So if you have an adverse drug response event in Spain and in Sweden, you don't want to be comparing them to individuals from France. You want to have a Spanish individual that you compare with a Spanish individual, a Swedish individual you compare with a Swedish individual. And in fact, they're actually doing this on a larger scale. So they wanted to have more of a global sample. I just looked at the European part of the data. Anyway, so it gives them a database of individuals that they can assume would not have the adverse drug response to compare the individuals who have the adverse drug response to. And then separately, once they find out what that variant is that causes the adverse drug response, they can look and see what its frequency is across Europe and say, look, this drug has no chance in Sweden, but it has a chance in Spain. Again, that's kind of presupposing that the differences are large enough. But that's the kind of idea of what inspired them. And so the samples went from the academic groups to GlaxoSmithKline. They genotyped it, gave it back to the groups, gave it to some analysis teams, of which I was one of, and then now the data is all publicly available via a database system online. Yeah.
So the drug company then did the SNP experiments, the SNP chip experiments.
That's right.
When they did the genotyping of these people around Europe, was there any, did they have to sign a release form? Was there any kind of ethical issues? Did they have to go through IRBs? Did they have to, I mean, what at the ground level? Knock on the door someplace, or they ask for volunteers? How did that work?
All right, so I don't know the details for any of these particular, because there was multiple academic groups, but the way these things usually work is that academic groups will have to pass IRB approval for their local university and have consent forms that, in this case, would have had to have said, we're going to be isolating your DNA, genotyping it, committing it to publicly available databases, and you have to be okay with that. And then the approval would be given by the university. Yeah. Yeah.
Samantha, I was going to ask you a question. Do you understand what John is saying, that if you have allelic frequencies that different, let's say among different populations, we've discussed this in the class, that perhaps one allele in a Spanish population might allow an enzyme to degrade a drug or something like that, so the individual might be okay with that drug, but a different form of that gene, another allele, might not work really well metabolically with that drug, and they'd have an adverse reaction. That's what we're dealing with. You understand that?
Yeah, we were talking about African Americans and heart disease medication.
Yeah, good. Go ahead with your question.
Okay, I was just going to ask, up there you have 197,000 SNP locations.
Yeah.
Like, is that really how many you looked at in each of these individuals, or is that, I mean, is that where their clusters are coming from?
Yeah. And
I'm just wondering, that seems like a lot.
Yeah, it is a lot. And in fact, we actually looked at about 500,000, and the 197 are the ones that pass a number of steps to kind of clean up the analysis. So
that's why you have the overlap, because some of these groups share maybe like half of the 500,000?
Oh no, they'll share. They'll share
a lot.
Yeah, yeah. We'll get. There are
So
very few.
The color variations are like only small amounts of SNPs that are different.
The color variations, like on
When
those
you're talking about
graphs.
the clusters, yeah.
Yeah. Yeah, I'll give you an example with this European data set of what the actual allele frequencies look like. What I did is I have a slide where I have just 25 random SNPs and what the allele frequencies look like in Europe. Okay. And but yeah, and it's an extreme amount of data, and that's actually a really interesting thing about the challenges and the bioinformatic challenges is that with this much data, a lot of the algorithms that we'd been developing break, and so you need kind of brand new computational computer scientists getting involved and new ideas for how to analyze the data that can actually run in a finite amount of time. In fact, running structure on this scale of data is very hard. So people either thin the data down, they just throw out SNPs. They say, we're only going to look at 10,000 of them because 100,000 is too many to run the program. Or in one case, you guys talked about the science and the nature papers. One group just threw out SNPs. The other group ran actually rewrote the program so that it could handle this much data. Yeah. Okay, and but one particular way of dealing with this much data is this method called principal components analysis. And the idea here is, I lost that pointer. I guess it must be in my pocket. Where is it? Oh, okay, thank you. Okay, so the nitty gritty, this is more like where my involvement starts, okay? Where you've got each row here is an individual, and to be compact, each genotype can be represented by a number, a 0, 1, or 2, where these are the 3 possible genotypes for a particular SNP, okay? And so this is a compact way of representing the data. We have, in this data set, 1,400 individuals, and we start with 500,000 columns, okay? Because every one of those SNPs is a column. And then, like I said, I boiled it down to 197 of those columns based on technical stuff that's not too important. But go ahead, Julie.
Do you only work with genes that have only 2 possibilities for each allele, or do you have more?
That's a great question, yeah. So in fact, these SNP chips only deal with loci that have single nucleotides where you have the 2 possibilities. Now, if the third one arose, and you don't know, I mean, the third one could arise, and it could be out there in this European sample, it's going to screw up the genotype calling algorithm. It's going to screw up the chip, because this chip is designed to, it's designed for biallelic loci. And that third allele will make the genotype call perhaps be wrong. So, and a bigger issue is when you get to this scale of data collection, there are, if you have very small error rates, say 1%, well, with 500,000 loci, that's a lot of mistakes, right? So, there are issues about genotyping errors that play into the analyses that you have to be very careful about. And having a third allele is one of them. But also, to give you intuition a little bit more to say about that is that at any single base pair in the human genome, there usually are only 2 alleles. And that's because this comes from more of the insights of population genetic theory, which is for any one base pair to become polymorphic, there has to be a mutation that happens there at some time point, right? And that mutation then is going to spread. And that mutation is either going to be lost from the population just by chance, okay? Like I might have a mutation on one of my germline that won't make it to the next generation, right? Okay, or it's going to spread until it spreads throughout the whole population, okay? And the time scale on which that happens is actually much faster than the amount of time before another mutation will arise at that site. So you go from having everybody the same to then having 2 variants. And you'll go back to having 1 variant before you ever have a mutation arise to make 3 variants at once. Okay? So it's really because mutation rates are so low at a single base pair that's what you see. Okay.
So you're not really losing much by not analyzing the ones with 3 or more?
Right, because there's so few of them. Yeah, that's right. And an exception to this is microsatellite loci, because microsatellites mutate much faster because it's all these slippage errors. And so there you see at any particular microsatellite locus a lot of alleles. It's kind of about the ratio of mutation rate to the rate of genetic drift, of how quickly mutations will drop out of a population due to chance. Okay. That's a great question. Okay, so so this data is massive. It's huge. You got, hundreds of thousands of columns, and you want to try and learn something about how Europeans are related to each other just by looking at that table. It's going to be really hard. You need to somehow boil it down. And so there's this method called principal components analysis, and the idea is that it's a reduction of dimensions in that it will squeeze this down to just one number per individual, okay? And so each of these numbers represents all of the data in a particular row squeezed into one number, all right? And that number is sort of chosen so that collectively all of these numbers will have a pattern of similarity that's similar to the pattern of similarity in the whole data, okay? And you can do this you can say, give me just one number that summarizes this data best. Okay, how about give me a second number? And then when you have these two numbers, you can, you can plot each individual on a two-dimensional plot. So here I can put this individual, I'm going to put them at 0.05 and at negative 0.08. So they'd be maybe right there, okay? And I do this for each individual. Albert?
I don't really understand what these numbers are representing and how you can get 2 per person.
Okay, so it's arbitrary how many you get. You can sort of choose how many you look at, and you'll just get more and more detailed of a picture. You can, if you go back and if you have 197,000 of these, you could get 197,000 of the numbers, and you would kind of be able to rewrite what the data is. Okay, but more specifically, imagine if I kind of put a big F, like a function, on this big row of numbers, and that's going to spit out one number. Okay, that PCA was a way of choosing that function, that black box, so that the numbers I get do a good job of representing the variation in the actual data set. Specifically, the functions are always a linear combination of all of these values. So we're going to take some constant times the 2, some constant times the 0, some constant times the 1, and then add them all together. Okay? So we're going to search across the space of all linear combinations of the columns for the linear combination that will result in the most spread in the PC1 values, the most variation. Okay? And that's going to be a good number to summarize the data.
So is it like every new PC column just a different constant that you're multiplying?
Okay, right. So yeah, it's a new set of constants in that linear combination. And in particular, it's kind of works in an iterative fashion. So what we're going to do is you say, what's the number that has the most, the linear combination that will give me the most spread. Then you're going to say, given these values, what's the next what's the number that will give me the best spread amongst the remainder of the data? That's not quite the right way to say it.
These are like all statisticians, They explain things with equations all the time. I'm teasing you. Rose, you got it? You're the mathematician. Explain conceptually. Go ahead.
All right. Great.
You with me, Dora?
Well, basically, aren't you just combining the variations of them using like a linear combination, like approximation, using different like statistical models, and you're finding like the ones with the widest spread and then kind of the next best one, if that makes sense?
Yeah, it's sort of, if you think about regression and how you draw a line through your data, and then you have the residuals, PC2 is kind of giving you the most spread amongst the residuals. Okay?
Jessica, you understand that, don't you?
I don't know what you meant by the residuals
Okay.
or the regression.
Yeah, so, or the regression.
Getting into the real
right, so
science here.
I'll go back to the whiteboard.
Ryan understands that stuff, don't you, Ryan?
PCA is really cool. It's also kind of hard to explain.
Yeah,
It's
absolutely.
from Davis.
Absolutely.
So weigh in if you have some simple explanations.
Okay, so this is say we have a data set where we just had 2 values, okay, for it, and there's some spread to the data. Okay, those are all points, and there's some kind of shape to it. Okay, and right now the data set has 2 numbers, right? And I'd like to summarize the data with 1 number. Okay, the number that's going to best describe the spread is going to be, kind of the biggest feature of this data is really how far each data point sits along this line, not this line or that line, right? And so PC1 is going to align itself along here, okay? And then we can think about the residuals. So when I talked about residuals, I actually was thinking about these. If now I kind of define the distance between any of these points and this central line, okay? We're going to have a new set of values, right? And if the data existed in like 3 dimensions, I would have, just as many variables as I started with, -1, right? And I would have moved from a 3-dimensional space down to a 2-dimensional space. And so then I can just replay the same exercise of what's the best line through that 2-dimensional space and get it. So I know that might
Does that
help
help you with the residuals? Points off the line.
Points off the line.
And
yeah.
how does that fit into a pattern? And then are there other points off the line? And do those fit into a pattern? And are there other points off the line? Does that fit into a pattern? And then how many patterns do you wind up with when you look at all of these different things, in a sense?
Yeah, that's great.
So if you're looking at this massive amount of data, and if you have a tool, in this case it would be a fancy computer that has some algorithms in it, how does that tool look at this massive amount of data that none of us would be able to make sense of with our eyes, and how many different, let's say, pots, how many different buckets, so to speak, did the data sort out to? And does it all sort out, let's say, for example, in John's example here, all the points exactly along that line? In a sense, that would be one component, in a sense. Or because there's variation in scatter, can you see a variety of different patterns? And so you're looking for patterns, and each principal component represents sort of a different pattern, so to speak. And not getting ahead of his story, in a sense, conceptually, you're using these data to try and say, are there different patterns with respect to populations in Europe or in different locales? And how can the data break down so one can say, my ancestry came from 2456, Yale Avenue in Cleveland, Ohio, as opposed to 3504 Tullamore Avenue, 3 neighborhoods off in Cleveland, Ohio, and can you separate that in those kinds of ways, in a sense.
Right.
Am I correct?
Right.
That's what it really is, without getting into the mathematics of it. Yeah. Albert. Was that right, Ryan?
Yes. It's tough.
So then, what does the graph represent then? If you're plotting essentially the same thing twice, right? Like
What do
each
you mean?
person,
Yeah.
you did the same thing twice, basically, to get two different numbers.
Uh-huh.
So I don't understand what the graph is representing. You're plotting like someone versus themselves. I don't know.
So,
Because if you had only one, like PC1, then you'd get all of those dots, but like on one line, so you wouldn't be able to tell the difference between one subset. So like there you'd get, if you just had PC1, you'd get 3 subsets, but then you wouldn't see the difference between those, and PC2 shows that.
yeah, except in this data set, PC2 actually doesn't have much meaningful going on. There is no real differentiation along it, yeah. But you're right, in many cases you need to go further. So it's not that you're plotting the same thing twice. You're plotting, think of it's a concept from data compression. You think about how, like, with your computer, you start with the really nice photo that you got off of your digital camera, and then you boil it down to maybe a JPEG that you're going to put on your web page, which has a lot smaller file size. In that step, you go from a very rich color space to maybe a much simpler color space. And that's essentially what's going on here. We're taking 500,000 axes of sort of description of a particular, we have 500,000 pixels, and we're going to boil it down to 2. Okay? So it's a very coarse representation. And actually, PCA, there's a connection between PCA and image compression and the actual JPEG algorithm, which is kind of cool. But the kind of demonstration, I was hoping to just explain this by giving you guys an example, but you guys are so inquisitive, which is great. The example here is that when I did this on this exact data set that's right here, you get 3 clusters, okay? And which is neat because you can't just see that staring at this data. And it's also neat because I simulated this data. It's not real data. It's data that I simulated, and I knew the answer, okay, because I simulated. I made my own little world, and I said, this is, we're going to have 2 populations and have some individuals that are a 50-50 mixture of those 2, okay, where maybe one parent came from one population, the other parent came from the other. And what do you see? You see that one population forms a cluster, the second population forms a cluster, and the individual are 50-50, lie 50 at a kind of 50-50 spot in between the 2. Okay.
So this is a question I've always had about admixture, is that if race and populations are a gradient, then how do you define an admixed population?
So there's a great point. It's really relative to like the time scale that you look back. And I was hopefully going to talk about that later on, but just to shortcut, individuals, as they spread out across the globe, may have been more gradual and smooth, but in recent history, we've had all sorts of interesting combinations, right? So I have, a mother from Uruguay, a father who's part Italian, part Irish, so that my particular genome represents admixtures of that are meetings between very different ancestries that are not, it's not a very smooth migration step.
Would like the sort of SNPs maybe of that admixture, would it match up with like a gradient somewhere else or something? You know what I mean? Like do these admixed populations have different genetic signatures than like the normal kind of place on the grid? Like if you were to take the sort of waypoint difference between like Uruguay and Italy, like would the person that intersects in the middle of that have a different SNP profile than like admixed?
Sure, yeah, absolutely. And we'll hopefully I'll give you a picture of what that looks like. Okay, so yeah. So that's a great, that exact question comes up, and we're going to completely talk about that in just a little bit. Okay, so the point is this method has been seen as this computationally efficient alternative to running those clustering algorithms, because if there are clusters in the data, it will produce clusters in the PCA plots. Okay, and then separately, I did some work to show that if there are gradients, it will show gradients, and it will even show that it will kind of arrange the individuals geographically. Okay, so then so it's a useful tool to visualize patterns of population structure. It's not a formal statistical method in the sense there's no p-values, there's no rigorous like hypothesis test. It's just a way of sort of compressing your data down so that you can plot it and look at it.